Welcome to EPG Padasala. This is Dr. S. Gayatri Devi, Associate Professor from the Department of Biochemistry, Biotechnology and Bioinformatics, Avinash Lingam Institute for Home Science and Higher Education for Women, Coimbatore. To introduce, neuron or nerve cell is a structural and functional unit of nervous system. Neuron looks more or less like other body cells and contains cytoplasm, mitochondria and other body organelles. Although it is different from other cells by two ways, neuron has branches or processes called axon and dendrites. Now, let us see about the classification of neuron. Neurons are classified into three different methods depending upon the number of poles, depending upon the function and depending upon the length of axon. First one, let us see about depending upon the number of poles. Based on the number of poles from which the nerve fiber arises, neurons are again divided into three types: unipolar neurons, bipolar neurons and multipolar neurons. Unipolar neurons are the neurons that have only one pole. From a single pole, both axon and dendrite arise. This type of nerve cell is present only in embryonic stage in human beings. Bipolar neurons are the neurons with two poles. Axon arises from one pole and dendrites arises from the other pole. Multipolar neuron is a type of neuron that possesses a single axon and many dendrites, allowing for the integration of a great deal of information from other neurons. Depending upon the function, again the nerve cells are classified into two types: motor or efferent neurons and sensory or afferent neurons. Motor or efferent neurons are the neurons which carry the motor impulses from central nervous system to peripheral effector organs like muscles, glands and blood vessels. Generally, each motor neuron has a long axon and short dendrites. The sensory or afferent neurons are the neurons which carry the sensory impulses from periphery to central nervous system. Generally, each sensory neuron has a short axon and long dendrites. The third type is depending upon the length of the axon, in which neurons are divided into two types: Golgi type 1 neurons and Golgi type 2 neurons. The Golgi type 1 neurons have long axons. Cell body of these neurons is in different parts of the central nervous system and their axons reach the remote peripheral organs. The neurons of the second type, Golgi type 2 neurons, have short axons and they are present in cerebral cortex and spinal cord. Now, let us have a discussion about the structure and functions of neurons in detail. The neurons are found to have organelles typical to many other cells, which include nucleus and mitochondria. It is made up of three parts, namely nerve cell body, dendrite and axon. This is clearly explained in the figure. It has one or two renowned nucleoli and it does not contain centrosome and hence it cannot multiply like other cells. The cell bodies, they are the small basophilic granules found in the cytoplasm of neurons and also known as nissal substances and tigroid substances. These bodies are present in the soma and dendrite but not in axon and axon hillock. Nissal bodies are membranous organelles containing ribosome and the function of it is found to be the same as rest of the endoplasmic reticulum and Golgi bodies for the synthesis of proteins in the neurons. In soma, the formed proteins are transported to the axon by axonal flow. During neuronal fatigue or injury, these fragmented bodies disappear by chromatolysis and the granules reappear after its recovery from fatigue or injury. Now, we are going to see about neurofibrils. Neurofibrils, they are the thread-like structures present in the cell body in the form of a network. It consists of microfilaments and microtubules. It provides a support and shape to the neuron. Thereby, they help in the transport of nerve impulse. Mitochondria is one of the nerve cell organelle which is responsible for the supply of energy in the form of adenosine triphosphate. Mitochondria also form the powerhouse of the nerve cell. Golgi apparatus. They are the membrane-bound cell organelle which is concerned with processing and packing of proteins into vesicles. We'll now move on to dendrites. The term dendrite comes from the Greek word dendron, meaning a tree. Dendritic branching is also called as dendritic arborization and dendritic ramification. And the overall shape and the structure of a neuron's dendrite is called its dendritic tree. The dendrite is a structure branched out in the tree-like fashion process of neuron, and it is branched repeatedly. It has the organelles in it such as Nissl granules and neurofibrils. Dendrite may be present or absent. If dendrite is present, which may be in one or more in number, and it helps to transmit the impulses towards the nerve cell body. In some cases, dendrites may transmit signals from the cell body and release neurotransmitters to affect other neurons, although dendrites are where the majority of the input to the neuron occurs. Most neurons have multiple dendrites and commonly hundreds of twig-like dendrites that cluster near the cell body. Dendrites are one of the two types of protoplasmic protrusions. It extends from a neuron cell body, the other being axon. Now let us see about axon. Axon is the long process of nerve cell. Each neuron has only one axon which arises from the axon hillock of the nerve cell body, and it is devoid of Nissl granules. It extends for a long distance away from the nerve cell, and the length of the longest axon is about 1 meter. Because of the absence of Nissl bodies in the axon, proteins necessary for the nerve fibers are synthesized in the soma and not in the axoplasm. The squid giant axon, which is specialized to conduct the signals very rapidly, is close to 1 millimeter in diameter, the size of a small pencil lead. Axonal arborization also differs from one nerve fiber to the next. The complex arborization allows for the concurrent transmission of messages to a large number of target neurons within the single region of the brain. At evenly spaced intervals, gaps in the myelin sheath nodes as nodes of Ranvier occur along the myelinated nerve fibers. The myelination enables an especially rapid mode of electrical impulse propagation called saltatory conduction. In axons, the demyelination causes the multitude of the neurological symptoms. The white matter parts of the brains are characterized by high density of myelinated axons passing through them and a low density of cell bodies of neurons. The cerebral cortex has a thick layer of grey matter on the surface and a large volume of white matter underneath. Most of the surface is filled with the neuronal cell bodies. Much of the area underneath is filled with myelinated axons that connect these neurons to each other. Now we are moving on to the organization of nerve. Each nerve is formed by many bundles or groups of nerve fibers, which is clearly illustrated in the figure. Each bundle of nerve fibers is called as fasciculus. The whole nerve is covered by a tubular sheath which is formed by a areolar membrane. This sheath is called epineurium. Each fasciculus is covered by perineurium and each nerve fiber is covered by endoneurium. The axon has a long central core of cytoplasm called axoplasm, and the axoplasm is covered by a tubular-like sheath-like membrane called axolemma. Axolemma is the continuation of cell membrane of a nerve cell body. Axoplasm along with axolemma is called the axis cylinder of the nerve fiber. Axoplasm contains mitochondria, neurofibrils and axoplasmic vesicles. Axis cylinder of the nerve fiber is covered by a membrane called Neurilemma. Nerve fiber is not covered by myelin sheath, described as the non-myelinated nerve fiber. Nerve fiber which is insulated by myelin sheath is called myelinated nerve fibers. The area where myelin sheath is absent is called as node of Ranvier. The segment of nerve fiber between two nodes is called as internode. Formation of myelin sheath around the axon is called as myelinogenesis, and it is formed by Schwann cells in Neurilemma. In the peripheral nerve, the myelinogenesis starts at the 4th month of intrauterine life. It is completed only in the 2nd year after birth. Before myelinogenesis, Schwann cells of the Neurilemma are very close to axolemma, as in the case of unmyelinated nerve fiber. The membrane of Schwann cells is double layered. Schwann cells wrap up and rotate around the axis cylinder in many concentric layers. Functions of myelin sheath is mainly faster conduction. Myelin sheath is responsible for the faster conduction of impulse through the nerve fibers. In myelinated nerve fibers, the impulses jump from one node to another node. This type of transmission of impulse is called saltatory conduction. The insulating capacity, myelin sheath has high insulating capacity. Because of this quality, myelin sheath restricts the nerve impulse within single nerve fiber and prevents the stimulation of neighboring nerve fibers. Neurilemma, axis cylinder of the nerve fiber is covered by a thin membrane called Neurilemma. It is also called Urilemmal sheath or sheath of Schwann. It contains Schwann cells which have flattened and elongated nuclei. Cytoplasm is thin and modified to form the sheath of Neurilemma, and one nucleus is present in each internode of the axon. Nucleus is situated between the myelin sheath and Neurilemma. In non-myelinated nerve fiber, the Neurilemma surrounds axolemma continuously. In myelinated nerve fiber, it covers the myelin sheath. At the node of Ranvier, Neurilemma invaginates and runs up to the axolemma in the form of finger-like process. Functions of Neurilemma. In the non-myelinated nerve fiber, Neurilemma serves as a covering membrane. In myelinated nerve fiber, it is necessary for the formation of myelin sheath, which is otherwise known as myelinogenesis. Neurilemma is absent in the central nervous system. So, the neuroglial cells called oligodendroglia are responsible for the myelinogenesis in central nervous system. Neurotrophins. Neurotrophins or neurotropic factors are the protein substances which plays an important role in the growth and functioning of nervous tissues. The initial growth and development of nerve cells in the central and peripheral nervous system. This indicates the possibilities of reversing the devastating symptoms of the nervous disorders like Parkinson's disease and Alzheimer's disease. Commercial preparations of neurotrophins are used for the treatment of some neural diseases. Neurotrophins act via neurotrophin receptors which are situated at the nerve terminals and nerve cell body. Neurotrophins bind with the receptors and initiate the phosphorylation of tyrosine kinase. Nerve growth factor was the first protein substance identified as neurotrophin. Now, many types of neurotropic factors are identified. The nerve growth factor NGF is a neurotrophin found in many peripheral tissues. It is a peptide with 118 amino acids. Each molecule of this NGF is made up of 2 alpha subunits, 2 beta subunits and 2 gamma subunits. Only the beta subunits have nerve growth stimulating activity. Functions of this NGF are they promote the early growth and development of neurons, and its major action is on sympathetic and sensory neurons, particularly the neurons concerned with pain. Because of its major action on sympathetic neurons, it is also called as sympathetic NGF. To conclude, the most captivating and complex system of the human body is the nervous system. For our appropriate interactions in the everyday life, millions upon millions of nerve cells are actively taking part inside our body. The nervous system includes the circuits that manage sensations such as pain and temperature, and circuits that provide our body to move and function. Neurons are ideal for muscles to move and digestive structures to carry food along through peristalsis, and they regulate breathing rate, heart rate and glandular secretions. Thank you.